taken after me. It's Friday.
Yes. All right. Hello, everyone. Welcome to the Thomas Expert Series. Thank you for joining us today. We're going to get started in just a second. Everyone, please double check, make sure your microphones are muted. This talk will be on demand within 24 hours, so please let everyone know how much you enjoyed the talk today and direct them to sign up for future talks. To get everyone a little familiar with the chat, let's see where everyone's tuning in from. I am in New Jersey. Jenny, where are you at?
I'm in Iowa today.
Iowa. I see some Florida, Philly, Indianapolis, North Carolina, Houston. Cool. We've got particularly South Jersey, not just New Jersey.
Oh no, they're two separate things. Oh,
they are two separate things. Alrighty, let's go ahead and get started. At this time, I'm going to turn it over to Jenny talking about You Bet Your Assay. How confident are you with your process controls?
All right. Thank you, Heather. Good afternoon and welcome to the Varizymes You Bet Your Assay webinar hosted by Thomas Scientific. I'm going to do a quick introduction and then I'm going to turn my camera off and I will continue. At Varizymes, we like to make the hard stuff easy by helping you solve problems in molecular diagnostics with a comprehensive range of solutions for enzyme and assay development. My name is Jenny Mayland, and I'm the sales director here at Varizymes. I have over 20 years of experience working in the food safety industry, as well as experience working in the preclinical contract research realm with medical devices. I'm here today with my counterpart, Brad.
Yeah, hi, I'm Brad Bellows. I'm the marketing director at Varizymes. I work in the headquarters here in Madison, Wisconsin. I'm responsible for sales collateral, customized stuff, anything for trade shows, and I'm also the shipping and logistics guy. So if you have any questions regarding packaging, lead times, couriers, things of that nature, I am your touch point. So with that, I will hand it back over to Jenny.
Fantastic. So we're going to go over our agenda before we begin the talk. I'm going to introduce Varizymes and what we do completely. Well, not completely, but you know what I'm trying to say. Then we'll do a brief overview of the new FDA final rule, the history of FDA oversight and the oversight of laboratories, medical, and devices. We'll review a few of the pros and cons of this rule. I'll go over the rollout timeline, and then we'll discuss some basic industry insights and responses. Then we'll talk a little bit more about detailed information on what general enforcement discretion is and the current legal landscape. Lastly, we'll talk about the solutions that Varizymes has to help you with in the near term and answer any questions you may have. So Varizymes, who are we? We're a business-to-business enzyme, protein, and reagent provider with decades of experience in molecular biology, genome mining, and protein purification. We like to take that expertise and create cost-effective processes that allow us to pass the savings on to you. Our product catalog features a wide range of proprietary enzymes, proteins, and reagents for basic and advanced molecular amplification. So on this slide, we talk a little bit more about what we do with our OEM services and things like that. I will let you go ahead and read that and take a look at it. But a couple of quick things I do want to point out. All of our enzymes and proteins are animal-free, and they are all available in glycerol-free and lyophilization-ready formulations. So let's move on and talk more about the FDA final rule regarding IVDs and LDTs. On May 6, 2024, the FDA issued its final rule amending its regulations around the in vitro diagnostic or IVD testing and its related subcategory, laboratory diagnostic tests, to include them as medical devices underneath the Federal Food, Drug, and Cosmetic Act. This rule phases out the FDA's general enforcement discretion approach that they've been utilizing for LDTs since 1976. Which means that the IVD manufacturers, including laboratories that manufacture tests to be used in-house, will be generally subject to the same enforcement as other IVDs and medical devices. That means that according to the ADLM in July of 2024, there are approximately 12,000 laboratories in the US that are eligible to perform LDTs. So that's a significant number of laboratories and impacts a significant number of people. This final rule amends the definition of IVDs at the 21 CFR 809.3A to make explicit that IVDs, including those manufactured by laboratories, are devices under the Federal Food, Drug, and Cosmetic Act. This amendment reflects that the device definition in this act and its IVD regulations do not differentiate between entities manufacturing the device. Accordingly, IVDs offered as LDTs are subject to FDA regulation as of May 6th. So let's go over the timeline history about FDA regulations and how it kind of meshes with this. So in 1938, the FFDCA passes. This allows the FDA the authority to regulate quality, safety, efficacy, and label accuracy of drugs and other products. In its original form, it does not mention medical devices. They weren't really a huge thing in 1938, but keep track of those things: quality, safety, efficacy, and label accuracy. In 1944, the Public Health Service Act passes. This actually enables the government to acquire new responsibilities, which include certifying medical laboratories. In 1967, the first Clinical Laboratories Improvement Act of 1967 passes. And this introduces licensing, regulation, and quality requirements for clinical laboratories that are operating across state or country lines. In 1969, we have the United States versus Bacto-Unidisc, which debates whether an antibiotic sensitivity disc designed for drug selection constitutes a drug or merely a device to determine whether or not such devices must be cleared by the FDA. SCOTUS ultimately rules that because the device contains a drug, pre-market clearance is required. Then in 1972, the FDA proposes to regulate in vitro diagnostics for the first time. Because it's a rapidly growing field at that point in time, on the basis that as medical devices, they'll fall underneath the Federal Food, Drug, and Cosmetic Act. The proposed rule covers labeling, performance, and reliability standards. So that goes back to the Act in 1938 when it was first passed. So in 1976, the Medical Device Amendment of 1976, or MDA, is passed, which explicitly places all medical devices underneath the FDA's jurisdiction and creates 3 risk-based classes and in order to regulate these, establishes regulatory pathways, and allows the FDA to ban unsafe devices. In 1977, the FDA publishes its final rule on medical devices. This rule exempts clinical laboratories from the MDA and also exempts devices intended solely for use by a physician, which is not generally available to or generally used by other physicians, including LDTs. And then in 1988, the Clinical Laboratory Improvement Act has amendments made to it. It becomes the CLIA of 88. And this defines regulatory requirements for all clinical laboratories, regardless of where they're operating, in order to include certification, accreditation, inspection, and standards for quality and proficiency. In 1992, the FDA drafts a compliance policy guide regarding tests labeled for research. It notes that some such tests are being used or commercialized for diagnostic purposes in this, and it asserts that such devices are subject to the same regulatory requirements as any unapproved medical device. The draft proposes a certification program, but exempts tests essential to public health. In 1997, the FDA publishes the final rule on analyte-specific reagents, which states that labs using LDTs are acting as manufacturers of medical devices and are subject to FDA jurisdiction, but declines a suggestion to classify all LDTs as a Class 2 or Class 3 medical device. In 2003, CLIA has another amendment to the 88, and this actually introduces additional requirements for LDT performance verification, excuse me, but places LDTs outside the FDA's purview by referring to methods developed in-house as test systems not subject to FDA clearance or approval. So you can kind of see where things start to get more murky. In 2014, the FDA drafts a guidance focusing specifically on LDTs. It declares that the agency no longer considers its enforcement discretion appropriate and proposes that labs who develop LDTs should be held to the same standards as commercial manufacturers of IVDs. In 2016, the House Committee on Appropriations directs the FDA to suspend its efforts to regulate LDTs, which, went for a very long time, a whole 4 years. In 2020, the Verifying Accurate Lead Edge IVCT Development Act, or VALID Act, is introduced to the Congress but is not passed. It outlines the FDA's intent to regulate all in vitro clinical tests, including LDTs. A couple more years pass, and the FDA announces a proposed final rule regarding to make it explicit that the agency has jurisdiction over all in vitro diagnostics, including when the manufacturer is a laboratory, and to phase out its discretionary approach to enforcement of LDTs. And then finally, the final rule is published this year on May 6, 2024, as well as 2 related draft guidance documents, one on enforcement discretion in declared emergency situations, and one on discretion in the absence of a declared emergency. Doing good? Taking a quick breather here? Think we're good? Now let's talk a little bit. Okay, let's talk a little bit about some of the pro arguments, the arguments for this new regulation. Again, we've got a bunch of things here on the screen, and we're going to review them. I've also got a couple other notes I want to make sure you guys are aware of. Right now, the CDC estimates that 70% of all healthcare decisions rely on laboratory tests. FDA regulation of LDTs would possibly expand insurance coverage of them, making it easier to use them when needed. Because they are not federally managed at this point in time, most insurance companies will not cover these, which becomes very important as we go into some of the reasons why we need them. The FDA firmly believes that there are public health concerns based on published scientific literature, peer-reviewed literature, lawsuits, their own review of the situation, and commentary on their proposed rule in 2023. The biggest thing that would change with this new regulation is right now there's no national database, and when a laboratory developed test is run, there is no accountability because we do not know who makes what test, and there are no records or national records to record any test concerns or failures. The FDA believes that this creates a heightened need for oversight of these tests, especially where test results are used by consumers to make potentially significant healthcare decisions without the involvement of a learned intermediary in a legitimate healthcare practitioner-patient relationship. That is actually FDA's response number 229 in the proposal list. The goal is to make sure that patients and practitioners are making decisions based on tests that have been validated and can be trusted. All of this indicates that the FDA has been exercising its enforcement discretion, but has been monitoring the situation. Since the original decision for enforcement discretion in 1976, the number of LDTs used in a human diagnosis has expanded exponentially. So instead of just being a one-stop, this physician creates it and it's just used solely there, it is now being utilized across state lines. Some of these are being commercialized and marketed to other laboratories, which makes it a much bigger deal. The fact that we don't have that national database, again, means that there's a massive lack of traceability when it comes to the tests. We don't know the ones that have a high failure rate. We have no ability to have a recourse if they impact the patient incorrectly, anything along those lines. So a lot has changed since 1976. A lot of these tests are being manufactured by laboratory corporations. So the whole landscape has changed. So what are some of the cons, the arguments against it? You'll note on the right side of this slide that's coming up that I have a bunch of group names, and these are some of the largest organizations within the clinical realm that help with testing or run the testing or are part of the laboratory grouping. There are a number of concerns that have been brought up. There were 6,700 different comments on the original proposed rule in 2023. But I pulled out just some specifics here that we're going to talk about. One of the biggest ones is there are concerns that the cost to be able to comply with these new regulations by the FDA will bankrupt smaller laboratories. Now, a thing to remember, the FDA is thinking nationally. It's thinking how it's going to save money, but still regulate in order to protect people. They're not necessarily thinking on the lines of small one and two people laboratories that are trying to run tests for people. So when the FDA is saying that it believes the benefits outweigh the costs and start throwing out numbers like the benefits of regulation will be 1 billion to 11.1 billion, and then the costs of regulating them are estimated to be 566 million to 3.56 billion. That's a great number to look at. You know, we've got billions versus millions, but it's overall, it's the whole nation. It doesn't actually address the idea that this is going to impact smaller companies very harshly. There's concerns that the FDA is duplicating oversight already handled by the Centers for Medicare and Medicaid Services and by the CLIA Improvement Act, or the CLIA Act of '88. However, in 2013, CMS and FDA provided a joint statement outlining which each one regulates. CMS, or CLIA, regulates the actual laboratory for the standards in quality testing regarding human and operations components. It does not regulate the actual performance of the laboratory developed test. It only regulates the operation, the performance ability of the technicians, their laboratory regulations, etc., to ensure accuracy and timeliness. The FDA is in charge of the actual oversight of the test devices or IVDs themselves to make sure they are testing exactly what they say they are. With the increasing scrutiny of the LDTs, there's a concern that patients will not have access to the tests they need to make accurate healthcare decisions. By increasing this scrutiny, we're basically saying we have to take all of these tests and move forward by putting them through the very strident regulations that the FDA requires, which is good in one respect, but it's going to limit patients in the meantime while that happens. With the increased regulations, there's also a concern that the ability to swiftly rotate and pivot, like in situations where we had COVID, where that ability to be nimble and create things on the fly was necessary, that the healthcare industry will not be able to respond as quickly. Also, many LDTs are innovated to help with rare conditions, things that don't necessarily fit into the larger overall model or large scale models of test development. So that would be rare conditions, underrepresented populations, and vulnerable populations. They don't fit into large scale development. There's not enough people with that. And then a changing or challenging regulatory environment will limit the motivation and the innovation, as the ability to create costs more and more money instead of keeping that broad and widespread, allowing more people to innovate and have the motivation to do so. Okay, so we're going to talk a little bit about the FDA responses. So the FDA took all of these comments by these larger groups and by smaller groups and created some responses. The College of American Pathologists, or CAP, officially opposed the final rule and then proceeded to work with the FDA to help make some changes. The American Hospital Association and the other groups that you saw also were part of this. But CAP really was proud of the fact that it helped the FDA align its risk assessments for the laboratory developed tests to be based off of patient risk, not the complexity of the test. So there's 3 tiers, 1 and 2 being low risk and medium risk, and 3 being high risk. Think of it as, if we're talking about cancer, think of it as, stage 1 cancer, stage 2 and 3 cancer, and then stage 4 cancer, or 3 to 4. So stage 1 cancer would be tier 1, 2 into 3 would be tier 2, and then 3 into 4 would be tier 3 as far as the risk assessment. These groups also helped the FDA establish a 5-step, 4-year rollout plan to phase out the general enforcement discretion. Discretion is included for integrated healthcare or laboratory systems. This means if you're part of a larger system like Mayo Healthcare or Allina or Health Partners or the VA, as long as that laboratory developed test is developed in that system, provided to the patient within that system, and then tested within that system, all within the same healthcare system, it will be grandfathered in and will not have to go through these regulations. Um, Basically, that allows some leeway for for patients that, you know, there's not a test made outside of that system for them, or if there is and it's FDA approved, it may not be approved for them in this specific situation. It allows for greater flexibility. And then there's some 1976 era LDTs where the general discretion will be continued. So let's talk about the 1976 era LDTs and other grandfathered LDTs. So when they're talking about 1976 era LDTs, we're talking about laboratory developed tests that meet the following criteria. They're 100% manual. There is no automation, there's no computer software, nothing. It is manual. It requires specialized technician training. So that technician would then be regulated by CMS and CLIA. The components of that test are approved for clinical use. And then the laboratory itself is CLIA certified for high complexity testing. So that's the 1976 era. But how does that all impact all the LDTs that are out there right now prior to May 6th that had been developed? Well, any LDTs that were produced prior to the publication date of the final rule on May 6th will be grandfathered in with this special or the general enforcement discretion if nothing changes. But as soon as a raw ingredient changes, a single step changes, or anything like that, they have to follow the new regulations. So it's limiting, but it still allows those tests to be run while their laboratories work on compliance. So you're probably thinking to yourself, what the heck is general enforcement discretion? Well, I can tell you definitely what it's not, and this is important. Enforcement discretion does not mean that your laboratory developed test is legal. It is not an exemption. In the eyes of the FDA, your laboratory developed test is illegal and in violation of the Federal Food, Drug, and Cosmetic Act. However, the FDA is choosing not to enforce or prosecute regarding that test at this point in time. The loophole here is the FDA can change its mind at any moment without prior notice. So that's going to roll into our next steps. Let's take a look at the legal scape and how it's going to look looking forward. The American Clinical Laboratory Association sued the FDA in April in an East Texas U.S. District Court claiming that LDTs are not devices. We are still currently waiting on how that's going and how that's going to move forward. A number of the other major groups and organizations that work in clinical testing and work with the technicians have put out statements opposing this rule. However, they're stopping short of lawsuits. The final rule has passed the executive branch, which means the president has signed off on it, but it is stalling in Congress. The Appropriations Committee did approve funding for FDA for the 24-25 year, but offered some precise advice on how to spend the money with an emphasis on not using it towards regulating with the final rule. According to the industry as a whole, the overall sentiment is to comply because it is a government requirement. As you can see, Sugath Pillai, the Global Medical Director of Oncology for Illumina, stated in an interview recently. So let's take a look at the timeline. It is a 4-year rollout. There are a couple of very important dates that are coming up. The first one, we already had July 5th, the final rule becomes effective. Now on May 6, 2025, these labs are expected to comply with medical device reporting, correction and removal, and complaint file requirements. This is the first step moving forward. May 6, 2026, there's a compliance to be expected in establishment of registration, listing your LDT or your IVD device, labeling, and investigational use of LDTs. May 6, 2027, quality management system regulation compliance is expected. On November 6, 2027, there should be compliance with pre-market review requirements for high risk, that would be the class 3 IVDs offered as LDTs. It is noted, you can see at the asterisks there, that you can be compliant by submitting it by that date. It does not have to be completely approved by that date. And then May 6, 2028, the compliance with pre-market and low-risk IVDs offered as LDTs. Again, you can be compliant by submitting a pre-market review application by that date. So the next slide kind of talks about some things that are closer or have been recently happened, some important dates. So we just had the August 22nd FDA hosted webinar to discuss how manufacturers of LDTs can comply with regulatory reporting. This whole webinar walks through how to comply and meet the needs by May 6, 2025 for all laboratories. We have another one coming up on September 24th that's going to talk about labeling requirements. The slide deck will be provided for you, so you can certainly use those links when we move forward. But the overall, the current environment is one where you, your customers, and third parties need to start assessing how they plan to move forward within the framework. We should prepare to comply with the applicable device regulations and make assessments on how to improve LDTs based on those upcoming regulations. So let's talk about what Varizymes can do to help. One of the main things that regulatory bodies require when it comes to point of care testing or end user testing is that your product is reliable, repeatable, accurate, and sensitive. Now, if you remember back to the very beginning of the talk, I pointed those out as part of the first act that got passed in 1938. This has not changed. I know it sounds like 4 different things, but it's all one grouping of things, and that's to make sure that your test works. Most people actually perform these tests in their R&D department while they're developing them, but they don't always comply with the regulatory standards that are required for verification and validation in the testing realm, in the commercial testing realm. You need to make sure that these standards are being met each time the test is used. This means based on the test that's being performed, the number of tests and any automation, and the ability of the technician to troubleshoot any concerning or inconclusive results, the number of controls that need to be in place will vary. We're going to talk more about controls in a little bit. It also varies based on the level of certification of both the laboratory itself and its technicians. As expectations of costs increase during this time, because having to start bringing all this stuff in, your cost is going to go up for creation, for compliance scaling. Varizymes can actually help negate some of those worries around costs when it comes to controls that are going to be needed in your tests and laboratories. I'll go more in depth into what controls are and how they are used and what Varizymes currently offers to meet these needs. When it comes to experience, as I said, Varizymes has over 3 decades of enzyme, protein, and genome mining experience, with thousands of citations accredited to our scientists and hundreds of peer-reviewed papers written by them. We leverage all of this capability to meet our customers' needs and bring forth cost-effective quality enzymes, proteins, and reagents. We also offer those OEM services, assistance with validation and verification, and research and development opportunities. We may be small, but we are mighty, and we make the hard stuff easy. So what is a control? We're going to start out at the top. A control is used to verify or validate that a system, assay, or technology is working as intended and to spec. You're going to hear the phrase process control a lot. A process control refers to a broader monitoring system that checks the entire experimental process to ensure reliable results across all samples happen. We're going to talk about 3 different portions of that process control system. We're going to talk about internal controls, positive controls, and negative controls. A thing to note, always verify what your regulatory body or certification body is going to tell you to do before you take any or make any process changes. Different levels of certification will require different frequency of use of controls and different timelines. So what's an internal control? An internal control is a control that validates that the system is working as intended. Specifically for molecular detection, we want to make sure that we're detecting something so that we know that the technology is actually going to do what we expect it to do, which is amplify either DNA or RNA. It's not necessarily the targeted organism or sample, but it may be a different organism that amplifies at expected times to verify that that test is working as intended. Typically, depending again on the level of your certification and that of your technicians, you can run an internal control. You can run one internal control per run on any specific machine. So for example, you're running an assay with 30 samples for one run in a single machine. You would only typically need one internal control in that run. So this one isn't necessarily the most cost prohibitive, but it does add up if you're running multiples of 30 samples. If you're running like 10 times 96 well plates, it can get very expensive. So what does Varizymes offer for this? We offer our MS2 phage. It's laboratory safe, non-infection BSL level 1 internal control. It's a wild type E. coli bacteriophage, which means it's non-infectious to humans. It is useful as control in RNA extraction or amplification assays, and it's nuclease resistant. This can be spiked into a sample for an internal control or just into a well next to the samples for that internal control. It's got a lot of flexibility and a lot of variation. Next, I'm going to talk about our cod uracil DNA glycosylase. It's a big mouthful, but again, it's important. It is, again, laboratory safe, non-infectious, and BSL level 1. But what this does, it helps with cross-contamination or carryover contamination in LAMP and PCR reactions. It removes the uracil from DNA, which it does it by substituting DUTP for DTTP in prior amplification reactions. But it is also usable in downstream applications, so it doesn't contaminate moving forward. It's a fantastic tool in order to help with any sort of cross-contamination, especially when you're doing automation amplification, where your sample tubes don't necessarily have a cover on them. So let's talk about positive controls. What is a positive control? A positive control is a control that utilizes a targeted organism or organism components to confirm that the test is set up appropriately to detect that target reliably and with accuracy. So this is a control that is literally saying this test is definitely detecting, I'm just going to throw out there, Salmonella, E. coli, COVID. We're trying to make sure that our test will actually pick it up. It can be either a whole organism or a targeted portion of that organism. Now, there's benefits and concerns to both. Concerns with the whole organism, it can be difficult to break open cellular walls. It can be difficult or time-consuming to purify. It can be expensive, and it can be dangerous for laboratory personnel to work with if there's no special equipment or a dedicated space. And even if it's an inactivated whole organism, we can't always be 100% sure that the whole sample is inactivated. Concerns with targeted portions. Targeting specificity can be complex. It's usually a little bit of a longer setup period, not with actually running the test, but creating that targeted portion positive control, because we want the actual DNA or RNA sequence. And the potential setup cost. Again, not the cost as you run each assay or each sample, but to get it set up and prepared. So what does Varizymes offer for this? We have the Varizymes VeriSafe. Again, it's a laboratory safe, non-infectious BSL level 1 control. These are engineered MS2 phage-like particles. So it has a nuclease resistant capsule on it. And we package RNA in there up to 1.5 kb of the target RNA sequence. So it protects that RNA from anything in the cell or anything in the sample until we are ready to actually break that capsule open. It can be spiked into various sample matrices. We do digital PCR quantification, and we do have already prepared sample types. So we've got various types of SARS-CoV. We've got foot and mouth disease, we've got HIV, and a few more. But this is also customizable. So if you have a target that's not on here, let's say you're looking for influenza A, we could definitely create that. So on the next slide, it just kind of walks you through how we would go about doing this. We would take our plasmid, do some transcription. We'd add the target sequence that the customer provides, do a little bit of translation, and then we're going to purify the viral capsid, and we're going to do some QC before we even send it to our customers. So essentially, this is a great way that's biologically safe for your laboratory technicians to utilize as a positive control. And then lastly, let's talk about a negative control. Negative controls are super easy. They are a simple vial of RNA or DNA-free water that is used to make sure that no environmental, situational, or cross-contamination occurs during that test run. That includes from sample prep to the very end. The negative control helps eliminate extra variables outside of the actual run as potential reasons for a false positive or a false negative. The number of times you have to use a negative control, again, will depend on your regulatory body or your certifying body. Depending on your certification, you may be able to run it one time a day, one time a run, or you may be required to run one per each sample line that you create. It should be always utilized, though, to make sure that aseptic technique is followed and environmental factors are not impacting test results. The number of times that I've seen airflow impact a test result for both molecular and microbiological testing is astronomical, because people don't think about that because it's just air. But a negative control can help you make sure that that could be a reason why your test didn't perform as expected. And again, please check with your certifying body on what frequency this should be used. That's all I have for you guys today. Thank you for coming and paying attention. And do you have any questions for us?
While everyone's typing out their questions, I want to take just a quick moment here and show you that some of the literature that was talked about today is inside of the Varizymes kiosk, as well as some web links and videos. And this talk will be on demand within 24 hours. So if you click on this on demand button up here, you'll be able to scroll through here and find it, as well as you could register for upcoming talks, and you could check out some of our labs. But I will hand it right back, and we'll see if we have any questions.
Fantastic. It looks like I see a few in here. Looks like Carl asked to address the gray area of discretion and how and when that is applied. Currently, the enforcement discretion has been applied across all LDTs, meaning that the FDA has decided until recently that they're going to just say all LDTs are just, they're not going to do any sort of regulation on them. Part of the reason that the final rule is happening is because that general discretion is so gray, and it's been happening, LDTs have blown up into this massive industry. That's why the FDA is doing what it's doing. I think that from what I understand and what I've read and what I've participated in regarding the FDA ruling and the LDTs, what it sounds like is happening is they are still allowing this general discretion for those grandfathered in LDTs for a short period of time, and then that's going to fade away. There is no current real, other than the timelines and, these guys get to be grandfathered in, but if anything changes, it's not. It's still very gray. There's really no guidelines around it, which is why I wanted to make a point of making sure everyone knew that the FDA could change its mind on it with a snap of its fingers. I hope that helps that answer that question. And then for internal controls, can they be endogenous or does it need to be target specific? They can be endogenous. They don't have to be specific to that target for an internal control. An internal control is basically just a control that says, hey, when we run this assay, when we run this product, we want to make sure that this internal control is saying that at this point in time during this, we are getting the reaction that we expect. So when you're looking at a real-time PCR or a real-time isothermal lamp, you're going to see the different arcs that come up on the screen as it starts to amplify. The internal control just has to be some sort of RNA or DNA related that will be broken open and amplified at the same time as your samples to make sure that the assay is running as expected. So it could be your positive control. It could be just an internal control. I've seen a lot of assays that run it based off of the positive control. So it could be one or the other, but if you don't want to run a positive control or you're not required to, you can just run that internal control. So Rhonda asked how exactly controls can protect point of care users and vulnerable populations. So what happens when you have an uncontrolled product of any sort and it's not regulated? So the controls, you're basically going to have so much variation in that one patient may get one result or one end of user may get another result, meaning that some of these smaller populations where these tests have no controls run to make sure that they are being run accurately, that they are actually testing for what they're intended to test for. So that would be the sensitivity and the accuracy. Their efficacy, meaning that every time they're going to pick up specifically what they're supposed to. For example, if we are testing for a UTI or an STI, there is a number of different bacteria and funguses that can actually cause you to have those infections. So the efficacy means that every time that test is run, we need to know that it has been tested for that. So there are different stations for those controls. Specifically, the ones that we're talking about for the LDTs moving forward is we want to make sure that we are running some sort of internal control, again, to make sure the test is actually performing as expected, which means that the customer or the patient or the vulnerable end user knows that it's going to be working and it's an accurate test that they're paying for, that it's going to tell them that their positive or their negative is correct. And that's what the internal controls are going to do. The internal control is going to say this test is working appropriately, it's working accurately. The positive control is going to tell you that yes, this test, even though it's working as intended, will in fact pick up what we intended to pick up. And that negative control will tell you as you're running that test that there were no outside contamination issues. Aseptic technique was followed correctly by the technician. We did not have any sort of touching of a micropipetter to the side of something else. We didn't sneeze inside it while we were creating the samples. We don't have a vent sitting right over our sample prep area, which can cause contamination, which itself, depending on what's floating around in the air in some of these laboratories, can cause a false positive or a false negative. So basically, it helps ensure that every step of the way we are testing exactly what we're looking for. It will save on patient emotional stress, because we're hopefully not giving them a false positive when we shouldn't be, or a false negative when we shouldn't be. And it'll save on money overall, because you won't be running retests all the time. I hope that answers that question. Beth asked, are the 1976 era LDTs the only exceptions to the rule? The answer to that is no. So again, within large institutions, health institutions, or clinics, as long as they are held within that grouping. So again, Health Partners, Alina, the Mayo Clinic, the VA, as long as they are created, utilized, and then practiced there within that conglomerate, those LDTs are grandfathered in, because they're all used in one entity and it's not outside of their realm. Anything that was created prior to May 6, 2024, as long as nothing in the formula or the process changes, they are grandfathered in and they are an exception. So they will be receiving general discretion. And then anything that is not automated, requires specific training, and is being practiced in a CLIA high complexity certified lab will be allowed to be exceptions to the rule. Will Duffel asked, can I clarify the difference between a lab diagnostic test and a lab developed test? That's me actually utilizing or flip-flopping words in there. LDT stands for laboratory developed test. I, in my brain, sometimes just say laboratory diagnostic test. A lot of your laboratory diagnostic tests are LDTs, but not all of them. A number of them are processed by larger corporations. They go through the FDA as a medical device. So specifically, to be clear, what I am talking about in this talk are laboratory developed tests that are utilized. I hope that helps answer that. Are there any other questions?
Not seeing anything else.
Sounds like a no.
No?
All right. Well,
Great. Thank you so much.
Thank you, everybody, and thank you, Heather and Beth, for helping us get this organized.
Yeah, thanks everyone for your time.
Have a great day.
Thanks, guys. Have a great day.